Hello everyone, welcome back to my channel. In the previous video, we were discussing about the general aspect of amino acid metabolism and we have discussed the transamination. In this video, we will discuss about deamination. So, deamination refers to the removal of the amino group from the amino acid. Fine. In transamination, there is just the shifting of the amino group, whereas in deamination, there is a removal of the amino group which will lead to the release of ammonia for urea synthesis. Release of free ammonia for urea synthesis. And the carbon skeleton which is left behind after the removal of amino group from the amino acid. This carbon skeleton is used for the formation of keto acid. And we already know the function of keto acid. Keto acid are used for energy production, used for the formation of glucose and fats, and it is used for the formation of non-essential amino acids. Whenever glutamate is involved, the transamination and deamination takes place simultaneously. Hence, the term which is used when glutamate is involved is trans-deamination. Fine. Now, the deamination is of two types. Deamination is of two types. The first one is oxidative deamination. The next one is non-oxidative deamination. First of all, we will discuss about oxidative deamination. In oxidative deamination, the deamination is followed by the process called as oxidation. It is clear from the name. The process deamination is followed by oxidation. It mostly takes place in kidney and liver. Mostly takes place in kidney and liver. The main purpose of oxidative deamination is to produce ammonia for the urea synthesis and to produce the keto acid, basically for the energy production. The first one is the production of ammonia, next one is the formation of keto acid for the energy production. Fine. Now we have to discuss the role of the enzyme called as glutamate dehydrogenase. Called as glutamate dehydrogenase. First of all, we have to know the function of this enzyme. What is the name of enzyme? Glutamate dehydrogenase. In transamination, the amino group from the amino acid will get transferred to ketoglutarate, alpha ketoglutarate, and this amino group will then get transported to glutamate. Then glutamate will undergo the process of deamination. The glutamate will undergo the process of deamination in order to form ammonia for the urea synthesis and alpha ketoglutarate. So this process is reversible. Fine. Now the formation of alpha ketoglutarate. The formation of alpha ketoglutarate from glutamate involve the formation of an intermediate involve the formation of a intermediate. What does this intermediate called? This intermediate is aminoglutarate. Aminoglutarate. This process is catalyzed by the enzyme called as glutarate dehydrogenase. Which enzyme? Glutarate dehydrogenase, about which we have to talk. So what is the role of glutarate dehydrogenase? Glutarate dehydrogenase catalyze the process of the conversion of glutamate into alpha ketoglutarate. Glutarate dehydrogenase can use NAD plus or NADP plus as a coenzyme. NAD plus or NADP plus as coenzyme. Fine. Now, how do we regulate this glutamate dehydrogenase? Basically, glutamate dehydrogenase. Glutamate dehydrogenase is a complex enzyme. Let me give some details about it. Glutamate dehydrogenase is a complex enzyme. It has zinc in it and it is present in mitochondria. It is present in mitochondria. Moreover, glutamate dehydrogenase has six identical units, each having the weight of 56,000 molecular weight basically. Fine. The activity of glutamate dehydrogenase activity of glutamate dehydrogenase is inhibited by basically this activity is inhibited by ATP, GTP, steroids and thyroid hormones. Fine. The activity of glutamate dehydrogenase is elevated or increased. Activity is elevated or increased with the help of GDP and ADP. GTP and ADP. Now, there is one noteworthy point. Whenever the energy of the cell is low, then the glutamate gets converted the glutamate gets converted into alpha ketoglutarate so that this alpha ketoglutarate can get used up in the TCA cycle for the energy production. Fine. So whenever this occurs when the cellular energy is low, when the cellular energy is low, then glutamate gets converted into alpha ketoglutarate so that it can be used in the TCA cycle. Moreover, when the protein consumption is high, when protein consumption is high, glutamate level also increases. Glutamate level increases and hence it will undergo deamination in order to maintain the equilibrium, and hence ammonia will be formed. Fine. Then, so we have read about one enzyme, that is glutamate dehydrogenase. Now let us read about amino acid oxidases. Amino acid oxidases. Fine. We know that there are two types of amino acids. There are two types of amino acids. One is the L-amino acid and the other one is D-amino acid. Fine. So, first of all, we have to talk about the L-amino acid oxidases. L-amino acid oxidases are the flavoprotein that have FMN as coenzyme. That have FMN as a coenzyme. FMN means flavin mononucleotide. The D-amino acid oxidases, D-amino acid oxidases are also the flavoprotein but have flavin alanine dinucleotide FAD, flavin alanine dinucleotide, as a coenzyme. The oxidases enzyme are separate for L-amino and D-amino acids. This you can conclude. Okay. Now this will perform the deamination process and form the ammonia plus alpha keto acids In this process, oxidative deamination, the oxygen is reduced to hydrogen peroxide. The body is protected from the harmful effect of hydrogen peroxide as the hydrogen peroxide is broken down by the catalase enzyme. The activity of L-amino acid is low in the tissue as compared to the D-amino acids. Why is it so? The activity of L-amino acid is low as compared to the D-amino acids in the tissue. Why is it so? It is so because the L-amino acid oxidases do not take part in oxidative transamination with in oxidative deamination when glycine and dicarboxylic acid is involved. Fine. So we conclude that the role of L-amino acid is less in the tissue as compared to D-amino acid. So we have to discuss about the fate of D-amino acid, like how they are dealt in the body. Look, the D-amino acid are not found in the body. D-amino acid are not found in the body. They are supplied through the diet. They are supplied through the diet, and they are even metabolized in the body. Fine. The D amino acid is found in plants and microbes or microorganisms. The D amino acid, when taken up in the body, the D amino acid, when taken up in the body, it undergo the process of transamination. First process, it undergo transamination in order to form alpha keto acid. In order to form alpha keto acid, this alpha keto acid will undergo the process of deamination in order to form the L amino acid. Then the L amino acid will will participate in the metabolic reactions. Or you can do it in this way, that the D amino acid will get converted into its respective keto acid, and then it undergo the process of transamination to form L amino acid. Right, you can do it that way, skipping the word deamination. I repeat, the D amino acid will get converted into the alpha keto acid. Then the alpha keto acid will undergo the process of transamination in order to form the L amino acid. The L amino acid will then take part into the metabolic reaction. The keto acid which will be formed is mainly used for the energy purposes or the precursor of glucose or fat synthesis. ठीक है. The D amino acid oxidation is very important. Look, the D amino acid oxidation is very important, or the D amino oxidases enzyme is very important, as it will initiate the first step, that is the conversion of D amino acid into alpha keto acid. It is the first step of the conversion of the D amino acid to the L amino acid. The enzyme which will initiate this first process is the amino acid oxidases. Now we will talk about the non-oxidative deamination. Non-oxidative deamination. In non-oxidative deamination, the deamination is not followed by oxidation. Is not followed by oxidation. The number one case that we will discuss is of amino acid dehydroxylases. Now, the amino acid which has OH group in them is serine, threonine, and homoserine. The amino acid hydroxylases will act on them in order to release ammonia and the respective alpha keto acid. Next is amino acid desulfhydrases. Desulfhydrases. In this, the non-oxidative deamination occurs with desulfurization. Desulfhydration. The amino acid which has sulfhydryl group in that is cysteine and homocysteine. Fine, cysteine and homocysteine. cysteine. With the action of amino acid disulfhydryl along with ammonia, H2S is also removed. Along with ammonia, H2S is also removed, and the keto acid which will form is pyruvate. Fine. Next, we will talk about the deamination of histidine. Next, we will talk about the deamination of histidine. The deamination of histidine. The enzyme named as histidinase will act on histidine in order to remove, like this is histidine. The enzyme histidinase will act on it to release ammonia and form the keto acid called as urocronate. Fine. So with this we will finish the deamination as well. This is all for today's video. Thank you for watching.